This video demonstrates the different steps in whole mount immunohistochemistry in chick embryo using HRP conjugated secondary antibodies. First, the embryo is fixed in paraformaldehyde. Then, endogenous peroxidase activity is quenched. The embryo is then incubated in primary antibody. After several washes, the embryo is incubated in secondary antibody conjugated to HRP. Color reaction is revealed using DAB substrate, and antibody staining appears orange. The advantage of this method over the use of fluorescent antibodies is that embryos can be processed for wax sectioning, thus enabling the study of antigen sites in cross-section.
Hi, I'm Delphine from the laboratory of Rick Finnell at the Center for Environmental and Genomic Medicine at Texas A&M in Houston. Today we will show you a procedure for performing whole mount antibody stainings in chick embryos using HRP conjugated antibodies. We use this procedure in the lab to study the morphology of embryos following treatment with anti-epileptic drugs. So let's get started.
To perform whole mount immunohistochemistry on chick embryos, first open an egg by tapping the shell with forceps and removing pieces of the shell. Remove the thick albumin with forceps and tilt the yolk sac with coarse forceps so that the embryo faces upwards. Next, use fine scissors to cut a square of yolk sac around the embryo. Remove the embryo from the yolk with a spoon and place in a dish containing PBS. Under a dissection microscope, carefully remove the membranes and yolk and transfer the embryo to a fresh dish containing PBS. After transferring the embryo, pin the embryo down with forceps and insect pins, and aspirate the PBS. Replace this with 4% PFA in PBS and allow the embryo to fix 1 hour at room temperature. To minimize potential microbial contamination, use dideionized H2O in all of the following steps. After the embryo is fixed, aspirate the fixative solution and dispose properly as chemical waste. Fill the dish with PBS. Next, using a microdissection knife, cut a square around the embryo to remove extra embryonic membranes. Remove insect pins with forceps. After the pins have been removed, use a blunt end Pasteur pipette to transfer the embryo to a scintillation vial containing PBT. Wash the embryo with PBT 3 times for 10 minutes each. Remove PBT from the scintillation vial and replace with PBTX, containing 0.3% H2O2, in order to inactivate potential endogenous peroxidase. Incubate for 2 hours at room temperature on a nutator. Wash the embryo with PBT, first 3 times for 10 minutes each, then 3 times for 30 minutes each. To stain the embryo with antibody, start by washing the embryo for 1 hour in blocking buffer, or 1% BSA, 1% NGS. We use NGS because the secondary antibody is raised in goat. Incubate on nutator for 1 hour at room temperature. After incubation, dilute the primary antibody 1:1 in blocking buffer and incubate embryo in this solution for 2 days at 4 degrees Celsius on nutator. The primary antibody dilution factor depends on the choice of primary antibody. Here, we use an antibody from the Developmental Studies Hybridoma Bank, which is provided as supernatant. We dilute it 1:1 in blocking buffer. Next, perform 3 10-minute washes in PBT followed by 3 1-hour washes in PBT. After washing, dilute the secondary antibody 1 to 2500 in blocking buffer, in this case peroxidase conjugated goat anti-mouse IgG, and incubate embryo in this solution overnight at 4 degrees Celsius on nutator. Perform 3 10-minute washes in PBT, followed by 3 1-hour washes in PBT. To develop the color reaction of the stained embryo, first perform 2 20-minute washes in tris buffer, 100 millimolar tris HCl, pH 7.4. Next, remove the last tris buffer wash from the vial containing the embryo and replace it with 5 ml DAB solution. Keep it in the dark on the nutator for 20 minutes. Dispose of the Eppendorf tip in the bucket containing a 10% bleach solution in order to decontaminate DAB. Add 16 microliters stock H2O2 to the vial containing embryos in DAB. Keep it in the dark. After 10 minutes, monitor the reaction under microscope. When the color reaction is complete, dispose of DAB in bleach bucket. Replace with 5 ml tap water. Next, perform 2 10-minute washes in PBS. To process for photography and wax sectioning, dehydrate the stained embryo in an ethanol series, 25%, 50%, 75%, and 100% for 10 minutes each. After dehydration, replace the ethanol with 0.5 to 1 milliliter cedar wood oil. This will make the embryo translucent. Process for photography in cedar wood oil. Following photography, return the embryo to the vial and replace the cedar wood oil with 100% ethanol. Finally, replace ethanol with 5% fast green FCF in 100% ethanol and incubate for 3 minutes. This step will make embryos visible for histology. Replace the fast green FCF solution with 100% ethanol and process for histology. Here are 3 examples of stained embryos. These stage 11 embryos are labeled with 15.3b9, or NOT1, a notochord specific antibody developed in the laboratory of Jane Dodd and Tom Jessel. After 2 minutes in DAB, the embryo is still transparent. After 20 minutes in DAB, slight labeling appears in the notochord. Finally, after 40 minutes, labeling of the notochord is complete. In this example, we show how PAX7, an antibody developed in Tom Jessel's laboratory, recognizes early stages of neural tube closure, as well as the cranial neural crest cells emerging from the midbrain. Here, the embryo is labeled with CROX20 probe from the laboratory of David Wilkinson, followed by NOT1 antibody. We've just shown you how to perform whole mount immunohistochemistry on chick embryos. This procedure can also be used on embryos which have been previously processed for whole mount in situ hybridization. In this case, briefly fix the embryo for 1 hour following in situ hybridization, and then proceed with antibody staining.
So that's it. Thanks for watching and good luck with your experiments.